So we will discuss about now bile acid. Okay, bile acid are detergents which emulsify lipids. Okay, bile acids are the detergents which emulsify the lipids. They have hydrophobic uh, and hydrophilic and in aqueous solution. Okay, it has the hydrophobic and the hydrophilic and in aqueous solution. Bile salt orient themselves around droplet of lipid forming micelles to keep the lipid droplet dispersed. Okay, bile acid are the detergent like which emulsify the lipid. Okay, they have hydrophilic and the hydrophobic and in a aqueous solution. Bile salt orient themselves around the droplets of the lipid forming micelles. Bile salt orient themselves around the droplet of lipid forming micelles to keep the lipid droplet dispersed. Okay, production. So, the bile acid are synthesized from the cholesterol by hepatocyte. Okay, they are formed from the cholesterol by the hepatocyte and excreted into the bile. Okay, they are formed from cholesterol. Okay, and they are excreted in the bile. Synthesis of new bile acid occurs as needed to replace bile acid that are excreted in the feces. Okay, and its synthesis is occur as needed to replace the bile acid excreted into the feces. The principal primary bile acids are cholic acid uh, and quinodeoxycholic acid. Okay, primary bile acids are the cholic acid and the quinodeoxycholic acid. They are made more soluble by conjugation with taurine or glycine in the liver. Okay, they are made more soluble by conjugation with the taurine and glycine. They become more soluble by conjugation with the taurine and the glycine in the liver. Okay, so principal primary bile acids are the cholic acid and the quinodeoxycholic acid. Okay, and they are become more soluble by the conjugation with the taurine and glycine in the liver. Okay, uh, enterohepatic circulation. So what is enterohepatic circulation? Enterohepatic circulation is of the bile acid excreted into the intestine. Enterohepatic circulation of the bile acids excreted into the intestine, about 95% are reabsorbed into the portal circulation by active transport mechanism in the distal ileum and recycled by the liver. Okay, 95% are reabsorbed in the portal circulation by the active transport mechanism in the distal ileum. Okay, in the distal ileum and recycled by the liver. Many of the bile salts are reabsorbed unaltered. Some are converted by intestinal bacteria into the secondary bile acids, deoxycholic acid, deoxycholic acid, and lithocholic acid. Okay, deoxycholic acid and the lithocholic acid, and then reabsorbed and small portion escapes reabsorption and is excreted into the feces. Okay, this was the enterohepatic circulation. Then what is the function of the bile acid? So the main function of the bile acid are emulsification of the lipid to increase the surface area available for digestion of the pancreatic lipase. Okay, emulsification of the lipid. Uh, okay, emulsification of the bile acids main role is the emulsification of the lipid to increase the surface area available for digestion by pancreatic lipase. Okay, it causes the emulsification of the lipid to digest it by the pancreatic lipase. Okay, then micelle formation to facilitate lipid transport and absorption. Micelle formation to facilitate lipid transport and the absorption. Then stimulation of bile products by osmotically attracting water. Stimulation of bile products. Okay, it will cause stimulation of the bile products by osmotically attracting the water attract the water. Okay, and electrolytes into the bile canaliculi. Stimulation of the bile products, it produces the bile, okay, which cause the osmotically attract the water and the electrolyte into the bile canaliculi, and the regulation of bile acid synthesis via negative feedback. This was the uh, function of the bile acid. Okay, emulsification of the lipid to digest it by the lipase. Then micelle formation facilitated lipid transport and the absorption. Then stimulation of the bile products to attract the water and electrolytes in the canaliculi, and regulation of the bile acid synthesis via negative feedback. Then, what is bile? So, bile is an aqueous alkaline greenish yellow liquid containing bile acid. Okay, bile is aqueous alkaline greenish yellow and liquid containing aqueous alkaline greenish yellow liquid containing bile acid, then cholesterol, phospholipid, bile pigment, electrolyte, mucus, and water. Okay, it will contain bile acid, bile pigment, okay, then water, electrolyte, mucus, and cholesterol and phospholipid. Okay. What is the function of bile? So bile functions to eliminate endogenous and exogenous substances from the liver. Okay, to eliminate the endogenous and exogenous substances from the liver, to neutralize gastric acid in the small intestine, neutralize gastric acid into the small intestine, and to uh, emulsify fats in the small intestine, okay, and facilitate their digestion and absorption. Okay, bile is the alkaline, so it's what it do, it do the it eliminate the endogenous and exogenous substances from the liver, to neutralize the gastric acid, okay, because it's acid, so alkali and acid will neutralize that acid, and it occur in the small intestine, and to emulsify fats in small intestine and facilitate their digestion and absorption. Okay, emulsify, it to do emulsification of the fat in the small intestine and facilitate its digestion and the absorption. Okay, facilitates its digestion and absorption. Now, production. Bile is secreted by the hepatocytes. It is isotonic and resembles plasma ionically. Okay, it is isotonic and resembles plasma ionically. This fraction of bile is called the bile acid dependent fraction. Okay, this fraction is called the bile acid dependent fraction. As it passes along the bile duct, the bile is modified by the epithelial cells. Okay, as it passes along the bile duct, bile is modified by the epithelial cells lining the duct by the addition of water and bicarbonate ion. This fraction is called the bile acid independent fraction. Okay, overall the liver can produce 500 to 1000 ml of bile per day. The bile is either discharged directly into the duodenum or stored in the gallbladder. The bile acid independent fraction is made at the time it is required during the digestion of the chyme. The bile acid dependent fraction is made when the bile salt are returned from the GI tract to the liver and is then stored in the gallbladder until needed. So, in the production of the bile, bile is secreted by the hepatocyte. It is isotonic and resembles plasma ionicity. Okay, this fraction of bile is called the bile acid dependent fraction. Okay, as it passes along the bile duct, the bile is modified by the epithelial cells cell lining the duct by the addition of water and bicarbonate ion. Okay, modified by epithelial cells lining the duct by addition of water and bicarbonate ion. Bile will be modified. The fraction is called the bile acid independent fraction. Okay, so this was the bile acid dependent fraction and this is the bile acid independent fraction. Okay, overall the liver can produce 500 to 1000 ml of bile per day. Okay, it will produce 500 to 1000 ml bile per day. The bile is either discharged directly into the duodenum or stored in the gallbladder. Okay, it can be discharged in the duodenum or maybe stored in the gallbladder. The bile acid independent fraction is made at the time it is required. Okay, when it's required, it will produce the bile acid independent fraction by adding the water and bicarbonate ion by the epithelial cell. Okay, uh, so that is during the digestion of the chyme. The bile acid dependent fraction is made when the bile salt are returned from the GI tract. Okay, so we have seen that when the bile acid independent fraction is made and when the so it's during the digestion of the chyme and when the bile acid dependent fraction is made when the bile salt are returned from the GI tract to the liver. Okay, this is from when the digestion during the digestion of the chyme, and this is when the bile salt returned from the GI tract to the liver and is then stored in the gallbladder until needed. Okay, and then it will be stored in the gallbladder storage. So, gallbladder not only stores the bile but concentrates it by removing non-essential solutes and water. Okay, it will also store and it also make it concentrated by removal of the non-additional solutes and the water, leaving bile acid and pigments mainly by active transport of sodium into the intercellular space of lining cell, which in turn draws in water, bicarbonate, chloride from the bile and into the extracellular fluid. Okay, so it do the active transport of the sodium and this will take the water HCO3 minus and chloride outside the in the extracellular fluid. Okay, within a few minutes of meal, particularly when the fatty food have been consumed, the gallbladder contracts and release the bile into the after fatty food. Gallbladder will be contract and release the bile into the bile duct. Okay, the sphincter of OD is relaxed. Okay, the sphincter of OD is relaxed, allowing the bile to pass into the duodenum through the ampulla of Vater. The gallbladder empties completely 1 hour after a fat-rich meal and maintains the level of bile acid in the duodenum. Okay, above that necessary for the function of cells. Now, regulation. The formation of bile is stimulated by bile salt secretin, glucagon, and the gastrin. Okay, the regulation of formation of the bile is dependent on the bile salt, secretin, gastrin, and the glucagon. Okay, the release of the bile stored in the gallbladder is stimulated by the, this is the formation of the bile, and this is the release of the bile. Okay, in the gallbladder, stored in the gallbladder, uh, that release of the bile is stimulated by the cholecystokinin, vagal stimuli to lesser extent, cholecystokinin, then vagus stimulation to the lesser extent. Okay, vagus nerve stimulation and the cholecystokinin.